Nano group of institutions becomes the next generation in the full range of academic, spiritual, social, and physical activities which make everyone sensible all-rounders. So today in this webinar, we are going to see about nano engineering for life. Nano engineering, which brings several traditional disciplines, including physics, engineering, and biology together. It is an emerging tool for manipulating major constituents of living organisms such as cells and tissues. The world is full of diamond and gems, and we are having some of them for today to build this international field webinar, Nano Engineering for Life. Let's start this session with a dialogue. Now let's start this webinar.
Ladies and gentlemen, very good morning to one and all. It's a great pleasure for me to introduce our speaker today, who is going to talk to us about the nano engineering for life. This is a subject in which we should all be deeply interested to get to know the latest development in the field of nanotechnology. Our speaker, although having spent almost a decade in his entire career in the research, especially in the field of nanotechnology, is an award-winning professional who has specialized in pharmacy for over 20 years. Ladies and gentlemen, please join with me in welcoming Dr. Mohan Kandhasamy, Senior Postdoctoral Researcher, Department of Chemical Engineering for Nanotechnology, University of Waterloo, Ontario, Canada. When I was preparing this introduction, I recollected him as one of the my best student during his UG program 15 years back. He's always spent time helping his friends and actively participated in classroom conversation. Even I must add my own opinion that he's truly a great person, a great scientist, a teacher, and a mentor. Over the above all, he came up in his life from an ordinary background to an extraordinary level with his self-supporting hard work. Now he made his community and his parents to feel proud to the core. Obviously, today we are here methodologies. Now, I move to explain about Dr. Mohan Kandhasamy's academic journey. Actually, Dr. Mohan Kandhasamy has completed his diploma in pharmacy from Directorate of Medical Education, Chennai, in the year of 2001. He completed his undergraduate and postgraduate program in pharmacy from the Tamil Nadu Dr. M.G.R. Medical University from 2006 and 2008 respectively. After that, he pursued his PhD in Republic of Korea at Gakon University under the topic of synthesis, functionalization, and hybridization of inorganic nanomaterials for bio applications. So let me express his professional experience in various fields. Actually, he started his career as a pharmacist, working part-time as a hospital and clinical pharmacist at KMCH Hospital, Coimbatore, India from 2003 to 2008. From 2008 to 2010, he worked as a lecturer and graduate thesis advisor at Nanda College of Pharmacy, Erode, India. Then he joined for PhD in Korea. After that, he Yangnam University as teaching fellow, graduate thesis advisor, and as an assistant professor from 2015 to 2018. From June 2018, as till today, he is working as a postdoctoral fellow in University of Waterloo. So apart from that, during his journey in the research, he learned a wide range of technical skills, especially the extensive experience in handling nanomaterials, lipid vesicle nanoparticle chemistry, synthesis of liposomes, engineering lipid, metal, nanoparticle composites, and anti-cancer cancer therapy. We able to design and conduct chemical synthesis, fabrication, and different characterization technique in inorganic nanoparticles. We have hands-on experience in bacteria, fungi culture techniques, and he experience in cell culture, cellular uptake studies, cell viability assays. He also trained in irradiation training, like trained in performing experiments with laser light for photothermal therapy, and he also enormous experience in the analytical techniques to operate the sophisticated equipments like absorption spectroscopy, dynamic light scattering, zeta potential, SEM, confocal fluorescence microscopy, HPLC, LCMS, etc. So in his resume, I could find a number of journals published. So he published wide range of journal and international level, which all are having very high impact factor of the journals. So apart from that, he has presented many presentations in international journals during his career. Finally, he is a peer reviewer in journals. So I will note few of the important journals materials: cancer, nanomaterial, molecules, current drug delivery, colloidal and surface biointerface. All this is serving as a peer reviewer, and also is consulting editor for International Journal of Nanomedicine. Also is editorial member for International Journal of Nanomaterials, Nanotechnology and Nanomedicine, and also working as serving as editorial member for Nanoscience and Nanomedicines. With this small introduction, I once again welcome our speaker, Dr. Mohan Kandhasamy, for this wonderful webinar session at PPG College of Pharmacy. I take this opportunity to thank the organizing committee members, Sorlin Selvo Joyce and Shyamala. I thank our principal, Dr. Vijayaraghavan sir, for providing me such a wonderful opportunity to deliver and introduce the wonderful speaker, Dr. Mohan Kandhasamy. I complete this speech with this. Thank you one and all. Over to Mohan Kandhasamy. Thank you.
Thank you very much for the long introduction, sir. I also just remembered our college days with you as a mentor and a hero to us, I should mention here. Yeah, saying that, I'm going to start my presentation. Today we are going to talk about, I'm going to talk about engineering nanomaterials for real-life applications. Today's agenda focuses the following parts. First part, I'm going to discuss about surface engineering of nanomaterials for antimicrobial therapy. Second part, I'm going to explain about sustainable nanomaterials for real-life applications. And as a last part, I'm going to explain about how to get ready for a research position or research studies in abroad. So these are the three important parts I'm going to cover in today's presentation. So moving forward, I'm going to talk something about nanomaterials and its introduction. So you guys are very well known about what are nanomaterials are, so I'm not going to deep into that, but I want to emphasize why these nanoparticles are important. So as shown in this figure,
introduction அவனோட பயோடேட்டா கழித்து அதனால அது முடிச்சிட்டு அவன் ஸ்பீச் ரைட்டிங் இருக்குது ನೋಡಿ ನಮ್ಮ ಸ್ಟೂಡೆಂಟ್ ಇಶ್ಯೂ ನೋಡ್ಬೋದಾ ನೋಡ್ಬೋದು
Hello, hello, Sorlin. Okay. Yeah. So the nanoparticles are so important because according to their size and shape, they provide different characteristics. So those characteristics should be tuned before deciding which application you are going to use it. So starting from carbon nanotube, metal sphericals, polymer nanofibers, dendrimers, and liposomes. So these are the different structures of nanomaterial. And the different shapes, yes, starting from sphere, tube, rod, plates, wires, and etc. And talking about the shapes, we should talk about the surface. So nanomaterials are like very smaller in size because of that they used to have large surface area than bulk materials. Because of that, they're kind of highly powerful, I should say. So those surfaces need to be optimized or tuned properly for its further applications, or else we could not achieve the intended purpose. So the surfaces could be modified using different techniques. As I shown in this cartoon, you can see hydroxyl group containing nanoparticles, how it could be modified, starting from making a 3D silicon network, attaching acidic group, attaching tertiary amine group, attaching halides, attaching thiol group, attaching chloride or porphyrins. Additionally, these nanoparticles further modified with the more biological molecules as shown in the right-hand side of the picture. So yes, nanomaterials has these much potentials. So you can attach one molecule on one nanoparticle or different molecules on different nanoparticles, or in the same nanoparticle you can attach different molecules. So it's entirely depends upon how we are going to apply. So that's why I mentioned nanoparticles are so powerful and it has more advantages than other micro capsules or bigger particles, bigger sizes of the particles. So after modifications, where are the places these nanoparticles could be applied? As shown here, it could be applied in different sections, starting from self-cleaning textiles. So those kind of materials can clean itself, means it will not allow any bacteria to attach, or it will not allow any dirt or dust to attach on the fabrics. So in that way, self-cleaning textiles are made. So that's the one of the potential application of the nanoparticles in textiles. Talking about biomedical industry, it's a very huge and evolving nanoparticle industry. I should say in my 10 year of this nanotechnology field, biomedical applications are evolving day by day. So they are so important, especially in this COVID-19 season. So nanoparticle surfaces are playing a major role apart from the uh, development of vaccine. Nanoparticles are playing the other side, like how to prevent a microbial attachment or virus particle attachment on the surfaces, or how to get rid of those kind of pathogens on the surfaces. So in that areas, nanoparticles are playing a major role. In next, food and agriculture, yes, again, food and agriculture these days they are using a lot of nanomaterials instead of like pesticides or fertilizers. So they are making it into nano farm, by which the plants can be fed with the minerals like copper or iron to have a better production capabilities. And in industries, yes, nanomaterials are being applied for making larger surfaces, like making parts for space rockets and all. In the case of electronics, most important part of nanomaterial applications is electronics. So the cell phones or mobile phones, whatever you're using these days, they are all made up of mostly with nanomaterials these days. They're starting from their display to their processor to their RAM. So everything is these days becoming nano and nano. That's why you are getting very big very big mobile phones to very tiny or very lightweight smartphones these days, including the laptops and the TVs. So these are the potential applications of nanomaterial in electronics. In the case of environment, again, nanomaterials playing a major role here these days, especially because of the global population and global warming. People are finding out a better solution to fight against these problems. So nanoparticles are playing a major role in how to remove the pollutants from water, especially in India. Indian soil and Indian river waters are highly polluted with antibiotics. So how to remove the antibiotics from the water resources? So there, nanoparticles are being applied and utilized. Again, in the case of energy part, the solar cells, as you all know, the sunlight is a very good uh, sustainable resource for producing electricity. So with the help of nanotechnology, those solar cells are being made and the currents are produced. In that cases, we don't need to depend on the rain or wind power anymore. So these are the potential applications of nanocomposites. So we in Gachan University, as shown here, worked on different gold nanoparticles. Gold is a biocompatible and inorganic material compared to the other inorganic materials, and it has more good biological properties and very less to no toxicity compared to the other nanoparticles structures. So we decided to go with gold. Again, gold nanoparticles, as shown here, they are available in different formats according to your synthesis method. You can synthesis spherical form, longitudinal nanorod form, nanostar form, nanoclusters, nanocells, and again nanoclusters, nanocubes, sorry. These are the different forms of gold nanoparticle you can prepare. Not only gold, any nanoparticle, but we were so interested towards gold because it has multiple properties. So we created gold nanoparticles and the applications of what we have made. So as shown in this overview or schematic, we first prepare the gold nanorods, which are like brown in color, and they are covered by magnetic nanoparticles. Those are kind of a dots in this blue color background picture. So those nanoparticles are mixed with the pathogenic bacteria like Escherichia coli and Staphylococcus aureus. Those are potent pathogens, and we passed the laser light to produce a heat. This method is called photothermal therapy. When the laser beam is passed to the gold nanoparticles, they start producing a heat according to the wavelength of the laser beam. So that heat could be used to kill the microbes. That's the kind of a hot topic in these days being used in cancer therapy. Sorry, in cancer therapy. So when the laser beam is passed, the gold nanoparticles convert the photons into uh, heat energy. So they produce heat. That heat kills the bacteria, and those nanoparticles, because of magnetic nanoparticle attachment, they could be reused. So we developed this one and we showed it could be reused at least 6 times. If fluorescent microscopic images of bacteria, and the right hand side picture shows the scanning electron after they got killed by the photothermal therapy. So this is the fluorescence microscopy image. So these are the different laser powers we use, starting from 10 millivolt to 130 milliwatt. So milliwatt is the very lowest power in the case of light energy. So we manipulated using these very small energy output. We create a live bacteria. So when the laser power is getting increased, all the green dots becoming entirely red dot, means every bacteria like gram-negative bacteria or gram-positive bacteria both are being killed at higher laser output. So in this way, we claimed our nanoparticle system is highly antimicrobial. It could be used as a in the water cartridges, like in India, you guys have Eureka Forbes, that kind of water cartridges, reusable water cartridges. So we were thinking about that, and we developed this system. So moving forward, the other part of this other section of this part is biofilm and its impacts on our health. So what are biofilms? Biofilms, as you all know or may not aware of it, biofilms are a group of bacteria which forms exopolysaccharides to create a kind of a layer on any surfaces. So this is the life cycle of the biofilms. When the bacteria are stick on a surfaces, that's called contamination. Then it started colonizing, like slowly growing its population, and additionally it will start growing the biofilm. This is called a 3D network. This 3D network is made up of biological proteins produced by those bacterial cells and polysaccharides, exopolysaccharides. So when time goes by, the bacteria grows and they become a very matured biofilm. That maturation part is the very uh, problematic part. Most of the biofilms are detrimental, like they're highly pathogenic. 80% of the pathogenic infection or bacterial infection are produced by the biofilm, especially from the hospital acquired uh, infections. So the matured form of the biofilm is so dangerous, so we have to do something to get rid of that. So in the other part, the matured bacteria will start releasing the potent bacteria to create the biofilm. So this is the kind of a cycle it's keep on going. So what are biofilms or biofouling again? When the bacteria sit and grow on any surfaces, they form these kind of a thick biomass which antibiotics cannot penetrate. So these biofilms are 1,000 times stronger than bacteria just in media or in water. So that's why the biofilms are the research on biofilms are so important these days. The right-hand side picture you can see, for example, how the biofilm causes the dental plague and the wound biofilm, as you all experience these ones. And biofilms on medical devices, this is really a serious issue. And biofilms on the pipelines on the other side of, apart from the biomedical applications. So our aim in Yangnam University was how to inhibit or how to remove these formed biofilms. So after the biofilm was treated with the anti-biofilm agents, it should become like the population got reduced almost 90 to 98%, and that way we can inhibit the biofilm formation, and that way we can get the solution for the biofilm infections. So as a part of that, we developed we were reviewing on different nanomaterials which are being used or applied in these days as a biomedical materials. So starting from the contact lenses, the contact lenses are covered by a layer of nanoparticles that could be used to prevent eye infections. And the nanoparticles are being used in the oral implants like filling or scaling in those purposes to prevent the oral infections from pathogenic bacteria, especially biofilms. And the endotracheal tubes, which are made by biodegradable polymers or tissue engineering materials. Those are the another high chances for biofilm formation. So these need to be treated with nanoparticles to have an anti-biofilm activity. Additionally, heart valves, urinary catheters, prosthetic joints, and wound dressings. So these are the other part of biomedical materials to be coated with nanoparticles to prevent the biofilm formation. So what are the mechanisms we can kill the or prevent the biofilm formation? There are different mechanisms using nanoparticles. First, starting from anti-adhesive nanoparticles, which could prevent the attachment of bacteria. That's the first way, but unfortunately, bacteria are prone to or like to attach on hydrophobic surfaces. So whenever the surface becomes more hydrophobic, the bacteria attaching and start manipulating over there. So these kind of surfaces need to be super hydrophobic. These days people are developing super hydrophobic surfaces where no particular dirt can stick on it. Next one, polymer grafted biocidal nanoparticles. These nanoparticles are created from polymers and it could be attached with some active compounds and they will be active against bacteria and using bioactive nanoparticles with the highly potent antimicrobial peptides or other biofilm inhibiting agents so the way it can kill the biofilm forming bacteria. And in the other types are reactive oxygen species or nitrous oxide releasing nanoparticles. These are the other ways are smart nanomaterials which could kill the bacteria on the surfaces. And as the last part, the another important topic is the stimuli responsive nanoparticles. These kind of nanoparticles can be get activated using sonication waves or laser light or UV light or visible light. So these are the other mechanisms where the nanoparticles can be used as an antibiofilm. So how we utilized this nanoparticle technology to create our antibiofilm system. So as a simple method, we used gold chloride and in ethanol in the presence of heat, it produced gold nanoparticles. These gold nanoparticles are highly active against the bacteria and as well as the fungi. Fungi are the stronger than the bacteria and they are more dangerous, like again 10 to 100 times stronger than bacteria. So these are need to be concerned. So we develop these bacteria, these cinnamaldehyde containing nanoparticles, and those are highly effective against the pathogens. So the above picture, the green one, the left hand side is the 3D confocal microscopy image of a biofilm, and after it get treated with the nanoparticle, it shows the complete removal of the biofilm. And bottom part, the when the fungi like Candida albicans or your yeast, the baking yeast, they have the tendency to create fibers when they get matured. So those are kind of more dangerous. So our nanoparticles can able to kill or prevent the fungi to show more pathogenicity. In these ways, we developed our system. So this is the one example for how we do characterize our formed nanoparticles. So left-hand side, as you all well experienced, is the normal UV visible spectroscopy method. So the blue line indicates the cinnamaldehyde curve. So the important distinction between these two peaks are you can see there is another peak at 520 530 nanometer. That's the uh, peak for gold nanoparticles. This is the peculiar character of gold nanoparticles. Gold nanoparticles have the capability to show different uh, UV absorption according to its different absorbance according to its UV according to its shape. So when the nanoparticles are spherical, they show a single peak at 530 nanometer. If the nanoparticles are a rod shape, then you will get the two peaks, one on here and one maybe around 700 nanometer. Anyhow, this UV visible spectroscopy confirms the produced gold nanoparticles are containing cinnamaldehyde also. In this way, we confirmed our first experiment and we move forward. See the right hand side image shows the transmission electron microscopic image. So you all are know about optical microscopy. So these electron microscopes are advanced ones. So to see what's happening or how the nanoparticles are formed, these electron microscopes are so important. So we use that one like and we characterize these nanoparticles. So these black color spherical shaped ones are gold nanoparticles, and this insert the magnified image shows there is a covering over the black dots. Those are cinnamaldehyde. So this way we confirmed again, okay, these nanoparticles are reduced by cinnamaldehyde and the cinnamaldehydes are attached onto the surface of the nanoparticles. So those nanoparticles are being used against different bacteria. You can see from this figure, starting from E. coli, P. aeruginasa, methicillin resistant Staphylococcus aureus. So these are the different strains we used against different concentration of our produced nanoparticles and cinnamaldehyde as control and gold nanoparticles produced by just chemical method as another control. Comparing to other two, our cinnamaldehyde gold nanoparticles are highly antibiofilm in nature. That results shown in the right hand side confocal microscopic images. You can see the dark like a floor mats of the 3D spectrum, 3D image of the confocal microscopic image of the biofilms. And after treated with cinnamaldehyde containing gold nanoparticles, they are completely removed. So whatever the type of bacteria, it doesn't matter how our nanoparticles are efficient, that matter. So we performed and we claimed our nanoparticles are highly anti-biofilm in nature. And additionally, we did cytotoxicity studies using the C. elegans as an animal model. This is a worm. So you guys are well familiar with the doing rats, mice or rats as an in vivo model. So the other possibilities are we can use worms as an in vivo model. So we developed using these worms as an in vivo model, and we checked our nanoparticles against ability. So you can see in the left hand side picture, the nanoparticle treated infected worms are getting cured or getting relieved from the infection when the days goes by comparing to the infected ones. So in that way, they are kind of highly anti-infective in nature. In this video shows how the nanoparticles got released from the worm after 7 days, or yeah, after 7 days. So this way we could confirm again our nanoparticles are not toxic, because it was uptaken by these worms and they got infected, they got cleared by the infection, and they can excrete the nanoparticles. So this way we claim our nanoparticles are safe to use. So moving forward, next part I'm going to cover is the introduction of nanomaterials, sustainable nanomaterials. Sorry, and its real life applications. What are sustainable nanomaterials? Nanomaterials which are obtained from renewable sources like nature sources are called sustainable nanomaterials. In University of Waterloo Department of Chemical Engineering, we are working on cellulose nanocrystals. So it's nothing but another nanomaterial obtained from plants. So this plant may be any plant as shown in here, any fruit or coconut or cotton, etc. But mostly these nanomaterials are produced from the tree bark or stem of the plant. So those nanoparticles as shown in here, they are derived from the plant cells. Usually sulfuric acid is being used to hydrolyze to get these cellulose nanocrystals. So these are the three different steps. At first step, the plant cells are degraded into form cellulose fibrils. So then again, they are hydrolyzed using sulfuric acid to produce microfibrils, and again after acid hydrolysis, we can collect the nano part of the fibers, leaving the amorphous region of the plant fibers. So this way, the cellulose nanocrystals are being made or produced. Here in our lab, we are procured the cellulose nanocrystals from a big company called Celluforce. Celluforce is the world's second supplier of CNCs. So we procure from them and we do all our experiments with that CNCs. So why cellulose nanocrystals are important? So cellulose nanocrystals are having very good or unique properties according to how you are obtaining those cellulose nanocrystals from plant sources. You can use sulfuric acid, you can use hydrochloric acid. It depends upon your experimental design. According to the hydrolysis, it will have the different surface groups like hydroxyl group, sulfate group, acidic group, or aldehyde groups. So, and again, it could be having cationic surface charge or anionic surface charge. In the terms of physical properties, it used to have a longitudinal structure as shown in here in this middle of this image, and it is more stiffer than steel. That's the one important good character of cellulose nanocrystals. That's why it's being used in different applications. And in the case of colloidal dispersion, it has its unique characteristic called biorefringence. When the concentration is changes, it reflects the light to produce different colors. So these are the unique properties of cellulose nanocrystals. So where are these cellulose nanocrystals are being applied or utilized? Since it's produced or obtained from natural sources, it's getting so famous these days. So starting from creating micro or nano films, conductive polymers, and transparent CNC films, Additionally, these could be used in developing smart materials like patches, solar cells in the case of electronic devices, foldable electronic devices, anti-reflective materials, biomembranes, smart paper, etc. These are the different applications of the cellulose nanocrystals. So, what we are doing with those cellulose nanocrystals? So, I'm just jumping directly into the our projects here, what we are working on, which are as a group or teams we are working on here. So, the first part will be the oral health. So, we are developing kind of oral remedy for antibacterial purposes again. So the oral diseases are the non-communicable diseases, and world is every year is half of the world's population is affected with the oral health. So why it is happening? Because there are so many materials already in the market as a replacement to be as a dental filler or dental resins, but the problem is they are not stable. They cause secondary caries like black spots on your teeth after filling, and these materials are being made with polymer. So after a long period, there is a problem of polymerization shrinkage, and these materials could damage your enamel, which is the hardest part in your body that's so important to protect your teeth. So these are the disadvantages. Additionally, amalgam is being used as a traditional material to to fill the oral cavities to prevent the infections, but amalgam contains high uh, amount of mercury, so which is highly causing neurotoxic, and it has poor refractive index too. So these are the reasons we could not use these already available materials. So what are the solutions we are providing? So we are developing here the center, the brown one is the cellulose, one single cellulose nanocrystals, and the blue lines are the polymers, what we are polymerizing, and the red plus symbols are quaternary ammonium groups, and the green dots are we are attaching plant agents like cinnamaldehyde, and the spherical uh, pink spherical particles are gold nanoparticles. So these materials are being created and these applied for different applications, starting from antimicrobial therapy. Yes, because of quaternary ammonium group, they are highly antimicrobial. And in photocatalysis, to degrade, as I mentioned before, in India, the water and soil is polluted with antibiotics, so that need to be removed or purified. Even though you can separate the antibiotics, but how you are going to degrade that molecule? So photocatalysis came into the place. So gold nanoparticles, in the presence of gold nanoparticles passing UV light, these kind of molecules can be degraded as a safer formation of CO2 and water. Additionally, these kind of cellulose nanocrystal modified structures could be used in imaging, cosmetics, and smart drug delivery systems, including produce coating, like your apple, all the vegetables could be coated by these cellulose nanocrystal to prevent its activities. So next part, this, this is our other important part. So what we are working on these days, so how to create a clean skincare products. Like, all of you guys are using a lot of cosmetics in your daily life, starting from just moisturizing cream to the different beautification products like serum, lip balm, lipsticks, and so on. So do you guys know how many chemicals you are putting on your skin every day? Minimum 200 chemicals. Yes. So those 200 chemicals are, most of them are synthetic. They are not at all good for your health, but you are being advertised and you are being used those things. So these are the different skincare products, starting from face cream, hand cream, sheet masks, and sun care. So these are the different products every day you guys are using. So these are all containing all the chemical ingredients. So according to the data, India is in number fourth place as the cosmetic consumer So India is the another cosmetic revenue producing country or a market, following United States, Japan, China. So India is producing lot of cosmetics in every day. So every of you guys are using lot of cosmetics. So every of you guys are knowingly or unknowingly putting a lot of chemicals on your skin. So it's kind of sounds scary, but that's the truth. Because of why there is no strong regulations like FTA for drugs. Drugs you cannot just use in the case of here in America, like in Canada, or if you go USA, you cannot go and buy an antibiotic just like that. So that's strict here they are. And the FDA are regulating a lot to prevent the make sure the produced or created molecules are so safer to the human beings. But in the case of cosmetics, the story is opposite, upside down. There is no proper regulation, so each chemical industry they can use whatever they want to prevent to create a better or shiny, attractive creams. So be careful when you are buying or choosing your cosmetics from next time. This is the one simple example. So sunscreens are being used for long. In India these days, people have started using a lot of sunscreens as compared to the olden days. But in case countries like Canada and the eastern parts like Japan or Korea, the UV rays will be intense in the case of winters. So our winter period is almost going to start from the end of November. So in those days, the UV rays will be very intense. There will be snow everywhere, so a small sunlight will reflect a lot. So the UV rays will be higher those days. So all the TV channels, government ask us to put sunscreens, even in summer too. So what happens, these sunscreens are having different materials starting from chemical UV filter, mineral UV filter, and nanoparticles and coarse particles. So chemical UV filters, yes, they absorb the UV rays and convert them into a better rays, which are not harmful to the health. And the other mineral filters are nanoparticles and coarse particles. So these nanoparticles just reflect the UV rays. So after you apply your sunscreen, what will happen? You just go back to your home and you wash off. So where the sunscreen or the ingredients will go? They directly go to your water stream like ponds, rivers, and to the sea. They will do. They will bleach the coral reef, and they will make some mutation or some deformity to the wild the sea animals. So that way, these nanomaterials are so dangerous these days. Some of the beaches like Miami, they already banned using sunscreens. So these days, people are kind of want to go with organic things. Again, so many companies, so many cosmetic industries, they are advertising their creams are organic, their cosmetics are organic, but again, they don't know about adverse effects and ingredient to ingredient interactions. Again, you can just take one organic formulation bottle like a cream, check its ingredients. Definitely, there will be more than 10 ingredients, a minimum. So they don't know, they will claim they have this property, that property, but they don't know what they will do. Anyhow, that story apart, they are developing very minimal naturally obtained ingredients to create a cream. So we developed a cream which are containing only 6 ingredients. Yes, only 6 ingredients. Those all are from natural sources and they are pretty stable. Still, we are developing. Soon we are going to launch our products in mini scale and we will send it out. So if you are interested, just give me an email. We will be in contact. We will send our materials soon. Okay, so those UV chemical filters, most of them are oxybenzone. Those are highly effective UV filters, but the problem of these oxybenzones are they are producing severe hormonal imbalances, especially to the woman. So that's way that from Waterloo Institute for Nanotechnology, we are developing very cellulose nanocrystal as a sustainable nanomaterial. So the next part we are in the other part we are developing probiotic delivery system. So what are probiotics? Probiotics are non-pathogens and they provide good health benefits and they are obtained from different uh, sources starting from yogurt, cheese, kimchi. So why probiotics important? So your digestive system is a second brain. Once your digestive system is healthy, your body will automatically work fine. Once if you it gets spoiled, every of your body organs will get spoiled. So that's why we need to maintain our microbiome where the probiotics grow, that is intestine. So most of the probiotic systems are developed like this way in the right hand side. So probiotics are encapsulated with prebiotics and they pass through the mouth, stomach, and intestine. There in the intestine, they proliferate, they grow more, and they produce health benefits. But the problem is most of the formulations available in the market, they got degraded in the stomach pH that is highly acidic. So our aim is to develop a probiotic system that goes all the way to the intestine and that will deliver the probiotics in the intestine without any degradation. So we are using cellulose nanocrystal and we have already developed two different types of uh, delivery systems, and again we are going to patent those things soon and we are going to produce them in a large quantities to sell in the market. So the other side, the marketed products, they use different layers, 3 to 5 layers to prevent the bacteria, but we are using very simple methodology without any harmful chemicals we are developing and we are achieving better activity compared to the marketed products. And the marketed products are expensive. We are trying to provide in very cheaper way. So these are the areas we are working here in University of Waterloo with sustainable nanomaterials for real life applications. So this is the end of the story of nanomaterials. Next, I'm moving on to how to prepare for a graduate study in a foreign country. So many of you guys, the studying or doing a graduation from a foreign country may be a dream. Yeah, so there are a lot of opportunities around. So how we are going to prepare this small part may be helpful for you. So just have a look, have a view about this. So first, fix your specialization. Since you guys are from soon, you will be a pharmacy graduate, so you have to fix your specialization. Which specialization you want to go? You want to go for pharmaceutics, or you want to go for pharmacology, or clinical practice, or something like that, and prepare yourself. So fix the specialization, start preparing yourselves towards your specialization. So preparation does take time and need energy, need patience. Most of all above, you need more focus. So do research on the field of study. So which uh, specialization you chose, again, there will be subdivisions in the case of research area. So which area you're going to start your research. Like in the case of pharmaceutics, there are different areas again.